So I guess the background for what I want to talk to you about is a system that is relevant not just in ocular melanoma, but in any situation in our body. So the immune system is what protects us against our environment. So it protects us against challenge with infections and through all manners of different infections, bacteria, viruses, parasites. And our immune system is really good at picking up things that are significantly different to the healthy bits in our body. And we've developed really smart ways of working out what is self, what belongs to us, and what doesn't belong to us, what is foreign. Now, the difficulty has been that in cancers, the vast majority of what makes a cancer is still very similar to the normal healthy cells in our body. So if you were to look at the genes in a cancer, maybe 1 or perhaps 2% of the cancer genes are different to the ones in the healthy cells. So that makes it quite difficult for our immune system to work out what is good and what is bad. And the other important issue here is that when an infection happens, you're well one day and you're ill the next day. So changes occur really quickly. And when a cancer develops, as you've heard from Professor Hyman, these changes can happen very slowly over many years and sometimes over many decades. So there are two things in the development of cancer that stand in the way of the immune system getting at the cancer. One is the changes are subtle, and the other is the changes happen slowly. So when cancer immunotherapy started to be developed, people said, well, this is never going to work because the cancers are just too similar to the healthy cells for the immune system to ever have a chance. And actually, what you're seeing is that the immune system hasn't worked because otherwise the cancer would never have developed in the first instance. And it's really against this backdrop that it is enormously surprising and for someone like me, very good and very fantastic that the whole field has changed really quickly over the last roughly 5 years. And it is now very much accepted that if you push the right buttons of the immune system, then the immune system is perfectly capable of eliminating even advanced cancers. And what we're learning at the moment is what these buttons need to be in order for that process to happen. So I'd like to talk to you a little bit about the experiments that have been done in people and the issues that we've learned about, so the successes that we've had. And some of this pertains to melanoma, even though very little yet so far to ocular melanoma. But also I think would like to talk to you about why things don't always work out and perhaps what we need to learn more about in order to overcome those hurdles. So 2009 was the year in which this all changed, and this was a year in which a number of randomized comparative studies of the flavor that Paul Nathan has talked about suddenly started showing that cancer immunotherapy can actually make a difference. And the extraordinary observation was that it wasn't one particular approach, so a MEK inhibitor, but a whole bunch of different cancer vaccines that all seemed to confer some benefit to patients at relatively low cost. Low cost in terms of side effects, not in terms of financial cost. And this was in prostate cancer and in melanoma and in lung cancer and in bowel cancer. And the approaches were really rather different. So very much like a MEK inhibitor and dacarbazine are very different beasts. The different vaccines were very different. So the learning outcome from that has been that there are many ways to a successful adaptive immune response. And it appears that there is not one single optimal way at the moment that we can identify. The next big story happened at the end of 2010, and that was when a drug called ipilimumab was first reported on in a randomized phase 3 study. So ipilimumab is a drug that switches on the immune system. In order to understand how this drug and its sibling drugs work, you need to know that when an immune cell encounters a virus, it becomes really geared up. So it's switched on, it's activated. And you need to imagine an immune cell a little bit like a sniffer dog. So the sniffer dog patrols through the tissue and looks whether it finds something that is abnormal. And if it does, it latches on. And this molecule that does latching on is called the T cell receptor. So it's a little bit like it takes a bite out of the cell that's abnormal. Now, when this interaction happens, then the T cell becomes activated, or you could perhaps call it excited. And that's a good thing because it makes the T cells kill the virus or bacterially infected cell. But it's a bad thing because if you've got the flu and you've got all these aches, then that's the net result of a T cell population becoming activated. So they make lots of molecules that make us feel lousy, they secrete cytokines. And if that goes on and on and on, that's a really bad thing. There are examples where that happens naturally. So there are bowel diseases, Crohn's or ulcerative colitis, where the immune system can't stop. And so our immune system has learned that there need to be really effective on switches through this T cell receptor, this latching on of the T cell. And then you switch the T cell on, but then you switch the T cell off again. So that, in other words, this activation becomes limited. These molecules are becoming better understood. And the first one that was really described in the United States by the group of Professor Jim Allison is a molecule that's called CTLA4. The name is perhaps not so important, but the message is that this is a molecule that switches immune cells off. So this engagement of the T cell, the sniffer dog in our immune system, with its counterpart, leads to activation and then the off switch. Now, cancers are really smart. They subvert these pathways. And Paul's already talked about how cancer cells subvert signaling mechanisms on the inside of cells. So genetic mutations switch molecules on that would normally come on and then become off again. So imagine it a little bit like a wound that needs to heal. So a cell that needs to fill the gap where the wound is needs to then stop growing when the scar has completed. Cancer cells don't stop. And so they don't stop growing, they don't stop moving, they don't stop dividing. And that's the net result of this is what we find when a patient develops metastatic disease. Lumps grow and then they damage the organ in which they grow. And ultimately, that's what we die of. So if the liver is replaced by growing lumps of ocular melanoma, then at the end of this, there is no normal liver left over. So what we're trying to do in oncology is to find molecules that reverse these switches. And Paul's talked about molecular switches, so that cut the wire on the inside of the cell so that, for example, the MEK pathway no longer functions. Immunotherapy thinks about this slightly differently. So what it tries to do is to not primarily target the cancer cell, but to target and activate the immune system that has no longer been effective. And so the CTLA4 inhibition, the net result is that inhibition is blocked. So a little bit like taking the brakes off the immune system is the net result. And when this was first tested, it was thought that, well, this was unlikely to work. And so it was with real surprise that these CTLA4 blockade molecules, the first one is ipilimumab, and I'm sure a lot of you have heard about that, and the commercial name is Yervoy, that these were tested in patients, then turned out to make a difference. So in skin melanoma, the outcome has been that roughly a third of patients benefit from this particular drug. And the real difficulty still remains that we have no way of picking out those patients and predict who will benefit. So far, we can, just like with the MEK inhibitors, just give it to everybody and see what happens. And clearly, we need to become much smarter at that. One of the problems with these drugs, aside from their cost, is that they do their job very well. So they activate the immune system. So the price that we pay, or that the patient pays, is that they are stuck with an overactive immune system. Now, if the immune system is just seeing the melanoma, then the net result is that the cancer is cleared. And indeed, there are patients who have been treated with these drugs who are now long-term disease-free. Long-term, I think the longest follow-up is about 15 years now. But the price then is that if you're switching on the immune system that was not completely clear what is good and bad in the body, that you're left with autoimmunity. And that means that there are normal organ systems that are attacked by the immune system. And the key molecule, or the key organ, is the bowel, so diarrhea, the skin rashes, the liver, hepatitis. But pretty much any organ system can be affected by this. Now we know that these adverse events are a sign, a biomarker, that the immune system has actually become activated. So when I see my patients and tell them you'll probably get a rash or you may get diarrhea, and that's not just a bad thing, but because it identifies the group of patients that is most likely to benefit. So what we can't yet do at the moment is separate out the attack to the tumor and the attack against the healthy system. Now, when ipilimumab and in skin melanoma vemurafenib came along, that was a revolution for a melanoma oncologist like myself, because I've spent about 20 years trying to find or put patients into trials, trying to find new treatments that would make patients better. So if you now are in a position where you can promise patients that there is a decent chance of benefit, even though in skin melanoma only about 30%, that's much better than everybody going wrong very quickly. But what we don't yet know is still how we can make that better and how we can pick the patients who will benefit. And there's a lot of work going on to try and work these things out. Now, I've told you about anti-CTLA-4 or CTLA-4 as a mechanism, but it turns out that there are probably a dozen or so, at least, molecules that have similar off-switch function in immune cells. So the next one that you've probably all heard about is a molecule called PD-1. PD stands for pro-death. So it's a molecule that was initially described as a molecule that was able, if engaged, to kill cells that expressed it. So it's a sort of getting rid of the dog altogether rather than just telling it not to eat its environment. But it turns out that, again, immune cells, when they're activated, upregulate not just CTLA-4, but also PD-1. And it turns out that PD-1 is particularly important for those cells that actually are the effector cells, the killer cells, the ones that get rid of tumor cells. Now, these cells are characterized by a molecule on the surface that's called CD8. And if you look at all immune cells, all the sniffer dogs that we have, then there are two big flavors. The ones make a molecule called CD4. They're the helper cells. And these helper cells activate other immune cells to do their job better. They're sort of the support army, if you wish. And then there are the CD8 cells, which are the cytotoxic or the killer cells. And on the cytotoxic cells, it appears that PD-1 is particularly important, whereas the CTLA-4 seems to be more important for the helper cells. So it was perhaps ultimately not a big surprise that when the first trial was done using PD-1 antibody, so again blocking the inhibitor, that the study was a remarkable success. And this was a study that was run in many different kinds of cancers, so melanoma among them, but also bowel cancer and lung cancer and renal cell cancer and kidney cancer and pancreatic cancer. And the real surprise was that for the first time ever, there was a drug that seemed to be working in many different kinds of cancers quite similarly. And for an oncologist who's sort of grown up on the you pick your poison according to the cancer that you're trying to get rid of, this is a real change in the way we think about it. Because, and I guess with hindsight, that's not so surprising, because actually the system that's trying to do the controlling is a generic system in the patient. You're trying to, after all, just switch on the immune system and not trying to get rid of primarily the cancer by killing the cancer cells. And so one of the things that we've learned in the meantime is that, therefore, PD-1 is a very attractive agent, and there are two of these that have made a bit of a splash. One is made by Bristol Myers Squibb, like ipilimumab, and the other is made by Merck, it's called Limovul. Anyway, I can't remember the name at the moment. They've made them all so complicated that it becomes increasingly more complicated. So it turns out, therefore, that these molecules are generically important. So for me, in talking to you in the context of ocular melanoma, I think this is really exciting because it gives us an opportunity to hope that this class of drug will also play a role in ocular melanoma. So we can't wait for the first trials to report on this, and there are a number of studies going on looking at both ipilimumab and the antibody against PD-1, which is called nivolumab, which is the BMS compound. A really big story came out of the question of what would happen if you have two drugs, each of which work a bit, so anti-PD-1 in about 40% of patients, anti-CTLA-4 in about 30% of patients. What happens if you put them together? And it turns out that that is doable. It gives you more side effects, but it seems to crank up the effectiveness to about 60% of patients in the cohorts that have been tested so far. So this is an extraordinarily big step. So if we had a group of drugs that use the immune system that work in more than 50% of patients, that would be an enormous step. So if you think about lung cancer, where some of these trials are being driven, then the best we can currently do with chemotherapy is to have a response rate in the sort of 30 or 40% range. So this would bring us beyond what we've been able to do in many solid tumors. And that's, I think, really quite remarkable. The other thing that we're learning, though, is that the immune system is continuing to fight back even when it's on the outside and as a net result apparently not won the battle. So when we look at cancers under the microscope, it is very clear that there are individual cancers that have lots of immune cells in them. And then there are other cancers of the same organ, so of the eye or of the lung or of the bowel, that have very few immune cells. And it turns out that patients with these two groups of cancers do very differently. So the first data set came out of a French observation by a chap called Gallon and Pagès, who looked at whether in about 500 patients with bowel cancer, you could work out who would do well or who would do badly according to how many immune cells there were. And it was really intriguing to find that in the patients with lots of immune cells, no one had relapsed at 5 years. And in patients with small numbers of immune cells in the tumor, most patients had died at 2 years. So there was a huge spread in the way the outcomes were. And that wasn't because the doctors were doing anything differently. It turned out that it was simply related to the number of immune cells sitting in the tumor. And that seems to again be a very widely relevant observation. So that's now true for breast cancer and again for lung cancer and for pancreatic cancer and ovarian cancer and so on. But the flavors of these individual cells that are sitting in the tumors that link with outcomes seem to be different. And there are a number of individual cancers where this observation may not be so straightforward. So when Professor Sarah Copeland reported on some of her observations some time ago, she told us that the tumors of the eye, when they have lots of immune cells, seem to do worse than patients whose tumors have fewer immune cells. So that seems to be very counterintuitive, and it is very important, though, because if this is a true observation, then we must be really quite cautious with the immune drugs that we might want to use. And it might not be so simple as simply using the drug that we've used in, for example, melanoma, where immune cells seem to predict for a good outcome. So I guess what I'm saying is that we need to look really carefully, and we need to really try and understand what's going on in the tumors in order to pick our drugs properly. So I think for ocular melanoma, what that means is that we must build a tissue bank of patients both with primary tumor and with tumors that come back that have metastasized. And we must take samples from patients whose liver has, for example, become affected by the melanoma, because only by looking at the tumors and looking at the recurrences will we ever be able to work out what is going on. The other observation comes out of a data set out of my own lab. We haven't published that yet. But when we looked at head and neck cancer, which is one of the cancers that is relatively common in particularly smokers and people who have drunk a lot of concentrated alcoholics, then there is a very clear difference between the genes and the proteins that are important for predicting outcome. And it turns out that in head and neck cancer, PD-1, the molecule that I've just told you quite a bit about, seems to be not very important. We can't find a lot of it. But another one of these 20 or so pairs, which is called LAG3, against which new drugs are also being made, comes up again and again. So the second important observation then is we must know from the tumor which molecules play a major role. Because if we are trying to develop rational trials, then there is no point using a drug that we think isn't going to work from the data that we already hold. So we must interrogate the tissues so that we can understand how we should pick the drugs. Because after all, this is not just another clinical experiment. This is a real life, these are real lives of real people that are at risk, and we must make our trials as smart as we possibly can. So I think the MEK inhibitor story is really clever, because here's a gene mutation, the GNAQ11 mutation, the GNA11 and GNAQ mutations that affect the vast majority of patients, 80 or so %. So targeting that is really smart. But I think when we start thinking about immunotherapies, we also need to become smarter. We shouldn't just do, as oncologists have always done, here's a new drug that looks exciting, let's try it in every single cancer that you can possibly think of, and then bring them together with every other drug. So if we think about, therefore, immunotherapy, I very strongly plead for collecting tissue as often as possible and then bringing that all together in the centers that have an interest, and that, of course, doesn't need to be Southampton. It could be Liverpool or Mount Vernon for all I care ultimately, so that we can start interrogating the tissues for making better decisions. However, when we think about cancer control, there is now also some really intriguing data that shows that this immune attack in cancers is an ongoing process. So there's a group in the United States that has asked this prospectively. They've taken mice that don't have an immune system and have made cancers, in this case a melanoma, and grown the cancer in the mouse. And then they've compared what the genes were that were abnormal in the cancer in the mouse without an immune system, and looked in parallel what the genes were that were abnormal in a mouse with an immune system. It became very clear that the immune system in the mouse that was immunocompetent did things to the cancer by getting rid of certain mutations. So in other words, there is a selective process going on that reflects the interface between the patient and in the patient, the sort of genetic makeup of the patient, the immune system, and the cancers. And this is a tripartite interaction where the tumor stroma and the tumor cells and the immune cells sort of battle it out in a three-way fight. And this is not a static event. And I think that that is really important, because if this is not a static event and if there's ongoing selection for tumor cells that are, because they're damaged, acquiring new genetic abnormalities, but at the same time are being selected for survival, because of course that's what the immune system does. It leaves over what it can't, it leaves behind what it can't kill. Then I think it also argues that we must try and do these interventions as early as we possibly can, before all this additional selection has happened. And Paul was mentioning that earlier on, talking, I think, Hannah, you were asking about the question of going into patients who have not yet developed metastatic disease, so adjuvant in the adjuvant context. And I think for immunotherapy, that is very much the way the field is going, to not wait until the immune system has failed and failed again, and until we've also ruined the immune system with chemotherapy and radiotherapy and whatever drugs we might throw at our patients, but to go into the setting when the immune system is healthy and when there's a much better chance that you could actually reinvigorate that in a way that might be important for the cancer. So we've looked in a small study with a vaccine. So my academic interest is in vaccinology, and we've made a vaccine that is a DNA vaccine in this particular instance, and we compared whether in patients with bowel cancer there was a difference in terms of the ability of the patient's immune system to respond, whether they had cancer or not. So we had two groups, patients who had no metastatic disease, so scans were clear, and then there was a group of patients who had metastatic disease, who had failed all previous treatments. And there was a very dramatic difference between the patient's immune system to do something useful with the vaccine. So 95%, or 11 out of 12 patients, That doesn't make it quite 95%, but anyway, 11 out of 12 patients were able to make immune responses to the vaccine if there was no cancer in the system that we could measure, if these patients were in the high-risk adjuvant setting. And only half of the patients were able to make an immune response if the cancer was present. So these were small numbers, but I think they illustrate the point that if you want to use immunotherapy, I think you're better off using it at a time when there is little cancer in the system. Nonetheless, even if things have gone badly wrong, the immune system hasn't failed completely. And there is some very intriguing data that would argue that even in those situations, you can make the immune system sit up and beg. So the data was originally described by the chap called Rosenberg. Steve Rosenberg is a surgeon at the NIH, and he had this idea that if you took tumor lumps and took the immune cells out of the tumor lumps and grew those and then give them back to the patient, surely you'd be giving back just the cells that are already trying to have a go because they're sitting in the tumor. And that turned out to be really rather impressive, and about 50% of patients responded to immunotherapy in that way. So this is called TIL therapy. TIL stands for tumor-infiltrating lymphocytes, and is a really good example that I think we have a long way to go in terms of developing our cancer immunotherapeutics. And in the UK, there's one centre in Manchester, Professor Robert Hawkins is doing that. There are a number of European centres that are developing these technologies forward. The difficulty is that you need lumps that are big enough. So most centers would say that you need a lump that's about 3 centimeters in diameter so that you can actually make enough immune cells in the laboratory and then grow them up. The downside also is that it takes some time, it doesn't always work, that you need to give chemotherapy to make the rest of the immune system make space for the new cells that you want to give back, and that these drugs that we have to use are quite toxic. But nonetheless, I think together with the data on the immunotherapies with the antibodies that I've spoken about and the T cells that are grown out of tumor lumps, I think this makes a very persuasive case that cancer immunotherapy is here to stay. And actually, it turns out that the big pharma companies also believe this. So Roche has just bought a really neat company in the United States. It's called Inovio, which makes DNA vaccines. And that's the first in class that has ever been taken up by one of the big pharma companies. So I guess if we were to look at this broadly, then one also needs to consider combinations of immunotherapies with tyrosine kinase inhibitors, such as MEK inhibitors, for example. It's clear that not all of these combinations go well together. So vemurafenib, the drug that's widely used in BRAF mutant melanoma, was combined with ipilimumab in patients with BRAF mutant melanoma and found to be really very toxic. A lot of liver toxicity limited the trial, and they had to stop after 4 patients. So clearly, if you push too many different buttons, and we not know what all the buttons are yet, then you potentially can run into real trouble. But I think conceptually, these ideas are really good. So it is likely that we'll see combinations of MEK inhibitors and immunotherapeutics. Whether that's going to be IPI or anti-PD-1, we'll have to see. And I think the final conclusion, and that's where I think I would like to stop, is that there's lots happening, and not just in the small molecules field, but also in the immunotherapeutic field. And my enthusiasm is also that this is a generic approach, because it seems so obvious that this is going to be true not just for skin melanoma, but for mucosal melanoma. That's early data now, and also for cutaneous melanoma, forgive me, ocular melanoma. But clearly, we need to do these early, and we need to know as much as we possibly can about the tumor at presentation, but also at recurrence, so that we can make our trials smart, so that we don't waste resources and patients' lives for something that is ultimately going to be failing. And so I think what we need your help with as physicians is to raise the awareness also of the physicians who are looking after you, to make sure that these collections are done. So, because I feel so passionately that only research can lead us to an understanding of the conditions, and it needs you to support that and to buy into that vision, because without this, we will never make any progress. Thank you.